And when I say we, I say the stem cell program. For those who don't know me, Alison Watry, together with Sylvia Evans, we are both co-directors of the UCSD stem cell program. And we always think about how to improve the communication, the networking, and we thought about teaming up with Evan, who has been running this seminar series for a long time. I remember when I was a postdoc here going to those talks and really taking advantage of the quality of speakers, the openness of the conversations, and really, I think, adds to my background on a stem cell biologist. So we now have the opportunity to invite people from outside, really provoke more cross-fertilization in our community. And Steve Goldman was one of the top people who people were excited to bring here, so invited. I'm glad he accepted. And his work is very interesting because it's quite broad. I think I start to pay attention to his work when I was doing adult neurogenesis, a little bit of how, songbirds actually create new neurons in the hippocampus. But then, he started to become more a glial person and really use gliogenesis as a model to study normal functions of glial cells, neurodifferentiation, the connections with brain cancer tumors, as well as how to use that as a transplantation or a perspective for therapeutic use in diseases that affect oligodendrocytes and glia in general. I'm particularly very excited that this can extrapolate in neurodegeneration. It's going to neuropsychiatry disorders as well. So I think this is what we're going to be hearing from now. So thank you so much, Steve, for coming. And please welcome Steve Goldman.
As Alison mentioned, I started working on neurogenesis a long time ago, and we were looking at neuronal production in adult canaries. It was something that Fernando Notabbaum and I found at Rockefeller a long time ago. And that led to studies in ascending species, if you will, until the point where we're looking at the adult human brain and the different types of stem and progenitor cells that were within it. And this is, some years back, maybe 15 years ago, we were sorting hippocampal neuroprogenitors and ventricular zone, and basically finding, hey, they're there in the adult human, but they're relatively scarce. In the meantime, we were just overwhelmed by the numbers of glial progenitor cells that we're pulling out. Using these extraction methods that were based on phenotype-specific transcriptional elements. And at a certain point, I thought, well, gee whiz, why am I working on neurogenesis, which may or may not, and that, as many of you know, is still controversial, may or may not be significant in adult humans, when what was really clear was that there were so many glial progenitors that this had to be significant. My clinical training is as a neuro-oncologist, and so this is the cell type of origin for gliomas, and I had an intense interest in that as well. And so putting all that together, the lab started to move more and more towards studies of gliogenesis, and as I'll go through today, all the different implications of essentially new findings in glial biology as they appertain to a broad variety of neurologic diseases. And so the disease list that one would normally associate with glial pathology is already significant, even before we start to think in terms of newer opportunities. And basically, that comes down to the myelin disorders. There are lots and lots of pediatric disorders of myelin, genetic and pediatric leukodystrophies, everything from the congenital hypomyelinations to the storage disorders that result in myelin destruction to the failure of glial progenitor maturation and expansion that occurs in cerebral palsy. All of these end up with hypomyelination or demyelination in kids, which can be variably either severely disabling or fatal. And in the adults, and every one of us here will, presuming that we live long enough, well, not necessarily become demented, but certainly have age-related white matter loss, which is one of the major causes of subcortical dementia. We think about Alzheimer's as being such an important clinical target, but almost half of adult dementias are actually based on white matter. disease as opposed to straight cortical dementias. And then the causes and the comorbidities with age-related white matter loss, diabetic, hypertensive, leukoencephalopathies. We're seeing more and more of this diabetic leukoencephalopathies now as people live longer and diabetes population becomes so much greater. It's really turning out to be a significant cause of adult white matter loss and then secondarily neurologic dysfunction, including dementia. But one disease that we all always think about as a classic myelin disease that is, of course, the focus of quite a bit of therapeutic effort is multiple sclerosis and its many variants like transverse myelitis or optic neuritis. So, and MS we're going to talk about in some detail. So it was with an eye towards potentially using glial progenitor cells as a treatment for multiple sclerosis. And I'm going to go into a little detail in terms of in what settings that it might be potentially used as treatment. But it was with that general eye towards establishing a remyelinating therapy for the adult acquired dysmyelination of MS that we started to take the purified glial progenitor cells that we were obtaining from tissues, and we're transplanting them into various animal models of demyelination to see whether or not the cells were capable of remyelinating demyelinated axons. So I'll just parenthetically at this point say that glial progenitors, there's a lot of terminology in the field that can be confusing. So glial progenitors can give rise to astrocytes or oligodendrocytes, the two macroglial phenotypes of the brain. This has nothing to do with microglial cells, which are the innate immune cells of the brain. So oligodendrocytes and astrocytes, in rodents and experimental models, there's debate in terms of the lineages, how restricted they are at different points during ontogeny. What is clear in humans, though, is that the glial progenitor is bipotential to generate either astrocytes or oligodendrocytes up until its very last division, and that is both in gray matter and white matter. It's about 5% of the cells in our brains are resident glial progenitors, and they are every bit as abundant and actually a little bit more so in gray matter than white matter. When we first isolated these cells and described them, we called them white matter progenitors, and then realized, hey, there's just as many in the gray matter. And they are actually a very interesting phenotype. They are not sitting there just to make new astrocytes and oligodendrocytes. They have functions of their own, which we'll get into. But in any event, we originally were calling them oligodendrocyte progenitor cells, which was the standard in the field at the time, until we realized that they were every bit as competent to make astrocytes. Now we just call them glial progenitors. So early in the game, we realized that, okay, here was a cell type that when we extract it from the human brain and then inject it, we can use it as a cellular vector for remyelination. But originally we were doing that from adult tissues, surgical resections. So then we thought, okay, this might be a feasible strategy to work up therapeutically, and so we started using fetal tissues. And this is late second trimester abortus. And these are medically necessary abortions in the setting of premature rupture of the membranes. And so the element of controversy that surrounds fetal tissue use did not so much appertain to these. But in any event, we developed the methods for extracting different progenitor populations from them, but especially the glial progenitor that was biased towards oligodendrocyte fate. And that was by selecting on a marker called A2B5 that recognizes a set of glial progenitor-specific gangliosides, and at the same time depleting the population of any neuroblasts using another marker, polysilylated NCAM. So that gave us a good cellular vector, and then we developed, without going into too much detail, a injection strategy for getting into the major white matter tracts of the mouse brain. And then when we injected the cells, we see this kind of pattern if we wait long enough. We went into the shiver mouse initially. In fact, this was based on some early work of Evans many years back, where he was using shiver and, as a model for looking at the myelination competence of injected cells. We realized, Evan had the right idea. This is a really good model to go into. And so we went in with this cell population and found that very quickly the cells were capable of migrating and dispersing throughout the brain. Now, shiver normally dies at about 19-20 weeks. These are mice that have an early stop codon on the first exon, the myelin basic protein gene, so they don't myelinate as the animals, they're born, they look normal, but as they're they get larger, as they get bigger. Axons become longer. Now they're not myelinated. There's more and more conduction failure. There's no capability of saltatory conduction in the absence of myelin. And then the animals become more and more symptomatic. They get ataxic. They start to have a tremor. This is why they're called shiver. They become weak in the hind limbs, the forelimbs. They go blind. They die. And that occurs like clockwork by 19, 20 weeks. And this already gives you an idea what's going on. This one we killed at 9 months. And basically, these animals are rescued. Every dot here is a human cell, though. And that's something we'll get back to. This is a single 14-micron section, so it gives you an idea of just how extensive the colonization of the brains are by the human cells. And they go ahead and myelinate, but this is all human myelin now. And that rescues the animals, and they go on to normal 2-year lifespans with actually what's most remarkable for a neurologist, a complete resolution of their neurologic symptoms. It takes a while, and so they do look sick, and they get worse and worse, but then they start to turn the corner, slowly get better until they are back to normal neurologically. But it is a slow process, and so these are matched animals looking at the midline of the corpus callosum, the major white matter tract of the brain. And so at 3, 5, 9 months of age, you can see the red axons, these are the mouse axons, becoming slowly myelinated by the green human myelin from the blue human oligodendrocyte, the progenitors that have now become oligodendrocytes. And it really is out to 9 months before it's complete myelination of the brains of these animals. So we wanted to be able to speed that up if we were going to be able to use this clinically. And so we defined a number of markers that were expressed by the cells at different points in their development, and identified CD140, which is a epitope of the PDGF-alpha receptor that comes on when the cells are still bipotential, but at that point relatively late in the game in terms of the number of population doublings they have to go through before they commit to terminal phenotypic differentiation. And so even though they're not biased one way or the other, they are only a few divisions away from becoming an oligodendrocyte or an astrocyte if they are put back in vivo. And so basically it's a way of speeding the process up. And when we sort it on the basis of the CD140 epitope, we see within 3 months we can get at least 50% of the axons myelinated by these CD140 sorted human glial progenitors. And so it's a way of speeding the process up. And that gives us also a means of identifying these cells in culture and knowing when we can use them for transplant purposes most effectively. And so it's in that setting we were looking at the RNA expression profiles of these cells during development and realized that we were getting enough information to be able to predict what ligands to be adding to these sequentially expressed receptors that the cells were expressing at various points in their ontogeny. With that information, we expected that we'd be able to essentially produce the phenotype from pluripotent stem cells. And so we first did that with embryonic stem cells and with iPS cells, and then essentially optimized the technique over a couple of years. As we learned more and more about the native developmental biology of the cells, we were able to apply that back to more and more refined production strategies. And so we ended up with this protocol that allows us to go from ES or iPS cell to glial progenitor cells with very high fidelity. It's basically, by the time we're out here, easily 90% of the cells are typically glial progenitors as defined by CD140 or A2B5. The 10% that are not are typically fibrous astrocytes, which actually seem to help the graft success. So that's the good news. It's an extremely high fidelity approach for making a given defined phenotype from a pluripotent stem cell. With higher efficiency than I think any other phenotype that's been described. That's the good news. The bad news is that it takes a really long time. So this is recapitulating human development, of course, and the oligodendrocyte is the last phenotype generated in the human body. And we don't even start to generate glial progenitors until 16, 18 weeks of gestation. It's not till about 28 weeks that it's really a rapidly expanding pool. And the cells don't really start to myelinate in the brain until about that point. Myelination is only 90% complete in humans at a couple of years of age postnatally. And of course, it goes on right through adolescence. And so these are very, very slowly maturing cells. So it takes about 140 days minimally. But in fact, we typically go out beyond 200 days in vitro before we have cells that are transplantable. But when we do transplant them, we get myelination from the ES-derived, or iPS in this case, glial progenitors, just as effectively, in fact, in some ways more so, than from fetal tissue. The cells will give rise to oligodendrocytes or astrocytes. These are adjacent sections. These are fibrous astrocytes that are essentially forming the infrastructure, if you will, for the glial progenitors that then line up along them and then go ahead and myelinate. And so you get this essentially longitudinal organization of the fibrous astrocytes in the corpus callosum with the oligodendrocytes on top of them and forming the myelin. Sheaths that then, of course, are surrounding the axons that are going through the same area. And so, effectively, this is the humanization of the white matter. The myelin that's generated is entirely normal, without going through all the individual details here. This is what human myelin normally looks like in all of its features, and it's entirely normal in terms of conduction velocities and functional acquisition of functional speed as well. So we end up with the ability to make it's essentially chimeric brains where the white matter itself is essentially humanized by implanted human progenitors. And we can do that not just from tissue, not just from embryonic stem cells, but from iPS, which also gives us the ability to do this on a patient-specific basis. And we've done a number of things with this as a model. First and foremost, still looking at effectively the biology of myelination and demyelination. We did this experiment. We wanted to see whether we could look at effectively the transcriptional events that accompany remyelination by human progenitors. This is a significant topic in therapeutic development in myelin disorders. If you have an area that demyelinates and now remyelinates, patients with MS, for example, will typically undergo a demyelinating episode in response to an immune activation, and then following a T-cell response and local demyelination, new glial progenitors will come in, divide, make new oligodendrocytes and remyelinate that focus. And that happens a couple of times, and at a certain point that process fails, and that's what then leads to chronic progressive MS after that process of progenitor-induced remyelination starts to essentially exhaust. And so we wanted to see what transcriptional architecture was being established by these cells, what genes they were expressing, and in what order during the process of remyelination, essentially of oligodendrocytic differentiation followed by myelinogenesis by human cells in vivo. Folks had done this for years in animal models. The information that had come out had turned out not to translate very well to humans. We thought, well, gee whiz, let's do this with human cells in vivo. And so we established these chimeric animals and then gave them cuprazone, which is a demyelinating toxin. We've looked at all sorts of demyelinating models, but this is the one that I think best replicates what's going on, at least in the pathology of human demyelination during the progression to progressive multiple sclerosis. And then we're able to take these cuprazone demyelinated brains and then allow them time to remyelinate from the endogenous, now human progenitor population. And one thing to remember here, not to remember, I haven't mentioned it before, is that oligodendrocytes are killed by cuprazone. Glial progenitors are not. So we've set up these chimeras. The myelin is there. The human cells are there. The progenitors are still there. They're demyelinated. Now the progenitors are activated, but they're human progenitors. They go ahead and remyelinate these axons that had been previously myelinated and now demyelinated. So it's different conceptually from axons that had never seen myelin before in their lives, like in the shiver. In this case, these axons were myelinated by the initial graft, and now they're remyelinating, and we're pulling these cells back out of the brains. And when we do that, first of all, when we take the white matter of these demyelinated, now remyelinating regions, and then run single cell on them, we see that we get very, very discrete clusters of still glial progenitors, human, and these markers NKX22 is upstream to SOX10, but they're very typical of glial progenitor ontogeny and phenotype. And then we look at essentially the whole population of glial progenitors that we're taking back out of these brains during the period where they are remyelinating. And that allows us to then look at, of course, the differential expression of genes by these cells as they are undergoing remyelination. And we can after network analysis, break that out into just a couple major categories. And what's interesting is that some were just as expected. So, for example, all these genes fall into the categories of thyroid hormone-dependent, retinoid receptor-dependent signaling, lipid transport, and these are the traditional targets, if you will, for inducing remyelination therapeutically from resident glial progenitor populations. And that is the trick, right? What we're trying to do is identify the targets for turning on endogenous progenitors to make them, at our command, induce new oligodendrocytes and remyelinate. We're trying to replicate the process that it's occurring in these animals. But at the same time, we see other modules pop up that were really not well understood. And so we're really picking up new targets. There are a number of members of the TGF-beta signaling pathway operating through SMAD4 that are represented here that turn out to be actually very viable targets, and also some downstream targets of TCF signaling, TCF7-like-2 signaling actually turned out to be quite upstream in terms of the potential induction of oligodendrocyte fate from glial progenitors. And just looking again at the functional classifications, just note down here, one of the functional categories that comes up looking at this module is schizophrenia. And that's something we're going to get back to. That there's some molecular homology, if you will, between an activated glial progenitor and some of the genes that are differentially expressed in schizophrenia. And we'll see that becomes an important point. But then still staying on the if you will therapeutic theme of how do we use these models to establish how best to have a remyelinating vector. We went and took the Cooper's-own model a bit further and took normal animals, absolutely normal, not shiver, not xenografted, not chimeric, and let them grow up at a certain point, then gave them a long-term Cooper's-own challenge. And typically, we give Cooper's-own for 4 to 6 weeks. In this case, for 20 weeks. And what happens is that the mice are basically demyelinating. Their own glial progenitors are starting to mobilize to make new myelin, but as soon as they start to become oligodendrocytes, they're killed by the Cooper's-own, and that just goes on and on and on. So basically, the mouse endogenous progenitors are maintained in a process of constant mobilization, and then their progeny are constantly killed. And after basically several months of this, the progenitor population starts to become severely depleted. And these animals, if you don't do anything else, will die. But what we do is transplant the human progenitors at various points in time during that demyelinating or right after that process of sustained demyelination. And the idea being to see whether in axons that had been normally myelinated and are now demyelinated in adulthood, if we transplant in human progenitors de novo, will they be capable of rescuing the animal and of course of remyelinating. And then we sacrifice to various points in time. And so it's a variation on the theme of the experiment before, but now in a much more clinically relevant setting. These are not neonatal chimeras. These are just normal mice that have the misfortune to see Cooper's-own and have their brains demyelinated. So when we transplant the cells, again, this is the CD140-defined population, they migrate abundantly through adult brain, and this is an important point. So we are capturing the cells specifically at a point where they would normally be migrating in development. And so what we're trying to do is capture the cells at a developmental stage that corresponds to that where they are typically undergoing migration from the medial ganglionic eminence of the developing brain out to the cortex and subcortical structures, where they will then integrate as glial progenitor cells. Well, that extraction of the cells based on CD140 allows that. And so these cells, they don't know they're in an adult brain, right? So they're not yet expressing the repulsive receptors that will essentially represent stop signals. Instead, they are behaving as they would in early ontogeny, migrating through the tissue. And it's not to anthropomorphize it too much, but not recognizing that they're in adult tissue. And they go ahead and then remyelinate this Cooper's-own demyelinated brain with absolutely normal myelin. Here's just one nice example of a human glial progenitor that has remyelinated its local surround. Now, to take this a little further therapeutically, one needs to be able to get the cells into and around larger brains. Even though the cells were clearly migratory, we wanted to know whether they would, whether that process would continue, whether they would migrate in larger brains effectively. We first went to squirrel monkeys and found that, in fact, they were both migratory and able to remyelinate focally demyelinated lesions. But we also realized that squirrel monkey brains are actually quite small. And so we went to aged rats, which have actually larger brains than squirrel monkeys. And for any of you either from Philadelphia or New York, especially ex-New Yorkers, the rats in the subway system can get the size of raccoons. And actually they continue growing through life, and the brains do too. And so it actually gives us a larger distance to work with. Anyway, this is Joanna Osorio's work, a pediatric neurologist in the lab who's interested in using this as a treatment for cerebral palsy. In her case, she injected aged rats 4 weeks, 8 weeks, 12 weeks, and 4 months after a single injection. This is one single injection in the corpus callosum of the glial progenitors. You can see the broad dispersal of the donor cells and, of course, the donor cell progeny. There's a lot of expansion that's continuing out to 32 weeks. Basically, by the time you get to just over a half a year, it's asymptotic, and the brain is fully infiltrated with the human glial progenitors, and they are largely replacing the endogenous mouse, which we'll get to. Now, the average migration of these cells is just over a centimeter. But they go up and down the entire neuraxis, and so down to the conus, the cauda equina, the entire length of the spinal cord from a single brain injection. It just takes time, and so the maximum distance traversed just in the rat is about 8 centimeters, and so we look at it as quite a migratory phenotype. We're doing peak studies now as per the FDA, but we expect that these cells will be every bit as migratory in all species. The limitations have to do more with the number of population doublings they're capable of than the actual migratory competence. So to be able to use these cells clinically, one needs to put them in or raise them under, I should say, good manufacturing practice compliance, which is GMP compliance. And to do that, we picked up a grant from New York State and built a GMP facility at Rochester, and then transitioned this technique that we developed for making the cells to the GMP environment, which it's one sentence, but it took us a couple of years. There is a bit involved in doing that in terms of having everything feeder-free and xenogen-free and completely defined reagents, et cetera. But in any event, it works fine. And so this is the technique that we submitted to the FDA last year. The GMP produced cells that are generated through this approach will myelinate the shiver brain just as well as anything else we've ever seen. And this is just the remyelination of a shiver using embryonic stem cell derived glial progenitors that are under GMP, and this is just a high power of the same, giving you an idea of just how dense the remyelination is using the GMP product. And so this is the kind of information we needed to be able to go to the FDA to propose this as a clinical trial for progressive MS. Now, the progressive phase of multiple sclerosis is an interesting one. A lot of folks will talk about using cell therapeutics for multiple sclerosis, but there are 2 phases of MS, and so you have to be very careful about which phase one's discussing. The normal early phases of MS, or the normally encountered, I should say. There's nothing normal about the disease process. But in relapsing remitting MS, the early phases of it, there's persistent episodes and recurrent episodes of demyelination, autoimmune demyelination, but they resolve. And then slowly over time, patients will become worse and worse as they have more and more remyelination failure after single demyelinating events. But then at a certain point, what we typically see is that most patients with MS will stop having recurrences like that. Okay? The autoreactive T cell clones probably have finite lifespans in the human. But then the patients will, and we call that burned out MS for whatever it's worth for in terms of just the clinical lingo. It's a slang way of saying that an MS patient has transitioned out of the relapsing remitting phase. But that doesn't mean that their disease has stopped. In fact, that's when it typically gets worse, and that's when they transition to the chronic progressive phase. By that point, a patient has lost a lot of myelin all throughout the brain. And axons require myelin. Axons require oligodendrocyte contact for survival. And so what happens is that slowly but surely axons start to die, and the neurons are lost, and both striatal and cortical atrophy starts to begin. And that's when patients will go from being functional to dysfunctional, from being able to take care of daily needs to being wheelchair bound and worse. And so it's a significant phase of the disease, and in fact, it's the least treatable. And there are lots of drugs now, immune modulators, that are quite effective in terms of cutting down the number of relapses, of suppressing the severity of relapsing or remitting MS. But by the time somebody ultimately transitions to progressive, then we don't have any therapies for that. And so the idea here is to use the glial progenitors to transplant them directly into the brain and hope for two things. The first is that the cells disperse and engage resonant axons the same way they do in the experimental models. And the idea there would be to preserve those axons that are threatened. These are axons that have been demyelinated, but they're still there, but they're going to die if they don't have achieved oligodendrocyte contact and enwrapment. And then, of course, the other purpose is to functionally remyelinate them and to restore conduction. But these are two independent endpoints. And one resulting potentially in the stabilization of disease, the other in actually the restoration of functions that have been lost. But these are two independent functions that we're hoping to see both, of course, play out. And so this trial, we went to the pre-IND originally actually with fetal tissue a couple of years back, and then realized that for scaling this up and for doing all the work that was going to be required in a clinical trial, we might as well just go straight to the embryonic stem cell derived. And so we went back to the FDA with another pre-IND almost a year ago. And now basically are just going through some of the safety toxicology assessments have to be done at an independent CRO to make sure that the cells aren't tumorigenic and this kind of thing. But assuming that comes back well, then I would expect that we'd be in the patients about a year and a half from now, given all the different timelines involved. So meantime, as this very therapeutic program was evolving, everybody involved doing it was starting to get more and more interested just in the nature and the implications of this degree of chimerization. I mean, when you look at so many human cells, you have to ask yourself, where are the mouse cells? And the answer is they're gone. So what's happening is that slowly but surely, the human cells are replacing the mouse within the glial populations. And so here we're looking at glial progenitors, human in green, mouse in red. NG2 is a marker of chondroitin sulfate proteoglycan type 4, which is expressed by all glial progenitors. It's they're human and mouse-specific epitopes. That's why we could stain for the same antigen, looking for the two different species derivations. And this is out at about 7 months or so, where the human cells were initially put in down here in the subventricular region, just below the corpus callosum neonatally. And now, months later, they've migrated out. The mouse cells are basically sequestered out to the cortical surface. This is what it looks like in essentially snapshots at 4 months, 8 months, 12 months. Ventricular zone surface, where the cells are first put in, out to the peel surface, these cortical strips, you see that there's an initial period of several months where the cells that have been transplanted are expanding within the presumptive white matter. The mouse cells are still all there on the cortex, but by the time you go out to 8 months, those mouse cells have really been mostly killed. Okay? And so they actually die in situ as the human cells are migrating out. Some of them actually themselves do migrate out dorsally, but by the time you're out at 12 months, there are no mouse glial progenitors left. And it's not just in these strips, in the whole brain. And so the human cells, as they are colonizing or replacing the mouse, the human cells have their own lives. We think of glial progenitors often, at least those of us working with them, as these little bipolar cells in vitro. And in fact, they're extremely complex cells. They're every bit as complex in terms of fiber complexity as one typically thinks of their larger cousins, the astrocytes. Actually, the astrocytes are not their cousins. The astrocytes are their daughter cells, because these are the cells that give rise to oligodendrocytes and astrocytes both. But this mapping here is just a neuroelucidant tracing on a number of glial progenitors the human glial progenitors in the chimeric brain. It gives you an idea of just how complex the cells are. They're always querying their environment. These processes are moving in the same way that microglial processes have been described as doing. And they respect one another's domains. There's a domain architecture with these where the fibers don't overlap, which mimics the domain architecture of astrocytes, which we'll get to, and yet the astrocyte and glial progenitor domains don't recognize one another. So it's an extraordinary degree of complexity in terms of the fiber organization of these brains. Now, over time, one of the things that glial progenitors do is replace astrocytes as they are lost. So oligodendrocytes are a relatively stable population in the adult brain. We lose oligodendrocytes slowly, and when we do, they are typically replaced, at least in the chimeras, and we believe it's the case in humans in vivo as well, by new oligos produced from the glial progenitor. But a much more significant source of glial turnover in the brain is that of the astrocytes. Astrocytes are always turning over. And when they do so, at least in the case of normal turnover, they are typically replaced by newly generated astrocytes from the resident glial progenitor. But now the resident glial progenitors are all human, so the new astrocytes are being generated from the human progenitors, so astrocytes slowly but surely humanize as well. And so here we're looking just at one snapshot of 7 months, and the green cells are human astrocytes, and the white matter by this point has largely replaced the fibrous astrocyte population by human cells, and now there's a lot of astrocytic infiltration of the cortex as well. And so the white cells are all human, but these cells have gotten to their destinations much later than these cells, and so they're on a different developmental program, but within the months thereafter, all of these will become astrocytes as well. And by the time you're out of a year, essentially all of the fibrous astrocytes in these brains are human, and most of the protoplasmics of the cortex are as well. So that has significant implications that we started to really consider, because human astrocytes are really different than those of lower species. Human astrocytes are much larger, much more complex. They have hundreds of fibers relative to mouse. These are at the same scale. This is a section of normal human adult cortex versus a shot of a mouse protoplasmic astrocyte. These are all stained for glial fibrillary acidic protein, the major intermediate filament of these cells. And so you see the mouse cell has a handful of processes and a little bit of fiber complexity. The humans are much larger. They have many more processes. And so individual astrocytes form domains. And so all of the fibers that are coming out of these astrocytes, except for the few end feet that go out to vascular surfaces, but most of these are encircling and encompassing synapses. And what happens is that all the synapses within a given astrocytic domain, a given radial domain, are essentially being set at the same gain in terms of their firing by the glutamate and potassium uptake thresholds that are essentially establishing what the environment is for excitability within that domain. And so these astrocytes are essentially coordinating information processing within the brain, because they are largely setting the gain for given synaptic events within their encompassed domain. Well, the mouse is doing the same thing, but the mouse, this given domain might have a couple of thousand synapses within it. The human is anywhere 10 to 100,000 synapses encompassed within that domain. And so, that suggested that human astrocytes are actually achieving much more information processing, or at least much more involved in it than their mouse analog. And that's of interest too, because evolutionarily, infraprimate mammals basically have small astrocytes, not very well developed. It's only when you get to the monkeys, New World monkeys, the astrocytes start to become a little more complex. Old World monkeys, significantly more complex, and a big leap in terms of the pleomorphism, how many morphologic types there are, as well as how much more complex they are when one gets to the great apes and humans. Now, in order to see what these cells might be doing in terms of contributing to the neural network, we then thought, well, let's look at these chimeras from the standpoint of the role and the function of the human astrocytes in the mouse environment. But in order to ask those kinds of functional questions, we need to know the human cells are really going to stay human, if you will, in terms of their organization and structure in the recipient mouse environment. Well, it turns out they do. If you tag them, as we did here with a genetic tag for GFP before at the progenitor stage before we transplant them, and then we look a year later, and the human astrocytes that have developed have very much mature human morphologies. And then we look at these essentially chimeric structures, And so this is an example of a hippocampus. The red cells are all human astrocytes out in the molecular layer. The green is human myelin. This was done in a shiver. And when we looked at long-term potentiation in these animals, we found it was greatly facilitated. And so that was our first hint that information processing in these brains really is different. And so that led us to ask this question, and this is largely Mike and Niedergaard's work, my long-term collaborator and wife. And what Mike did in this case was ask this very basic question: are these animals smarter? And we looked at a number of behavioral tests over the years. After we first published this work, but this was the first model we used, which was take the chimera versus the normal, and then look at the auditory fear conditioning. Basically, the animals are exposed to a shock after a prior exposure to a sound. They learn to associate the shock with the sound. But if it's at a relatively low intensity, they may or may not notice. They may or may not make that association. A smart mouse makes the association. A mouse that may not be the sharpest card in the deck may not make the association. And so we use that as essentially as a standard screen to see whether or not there's a difference in the cognitive capabilities of the mice. And when a mouse is scared, in other words, if it has made the association of sound to shock, and a sound comes on, if it's made that association, it gets scared, it freezes. That's what mice do when they're afraid. They just freeze. You can quantify the amount of time they spend frozen after hearing a sound, and that basically is a measure of their degree of anxiety and inferentially the degree to which they have made the association between the conditioned stimulus. And then we look at how that plays out quantitatively, and we see that the chimeric mice spend much more time frozen. All the mice in this series received one shock once after a sound. They hear the sound in their chamber. They get shocked right afterwards. Subsequent days, they're thrown into the same chamber. They don't get a shock. They're simply presented with the sound. And the chimeric mice spend much more time frozen, and then day after day, it's actually reinforced. The unengrafted control mice, they don't make that association. They're not particularly worried about it when they hear that sound. And what's interesting here is the mouse allograft controls, and so these are mice that received mouse glial progenitors at birth to be chimerized essentially with an allograft. And they don't make any specific association between sound and shock either. So it's just the human glial chimeras that do. So this was just one of our first examples. We've looked at novel object and place recognition, Barnes maze testing, a variety of largely Pavlovian tests as well. And these animals are quite superior in terms of cognitive capabilities. And so based on that, we asked, okay, if the human glial cell really is more evolved than its lower species counterparts, then we may be able to map diseases that are human specific, because now we have a phenotype that, for all practical purposes, is human selective, at least in terms of its degree of evolution and degree of network function. And so then the question becomes, what diseases, right? What neurologic diseases are human specific? And the neuropsychiatric disorders are, of course, at the top of that list. Schizophrenia, autism, the autism spectrum disorders, bipolar disease, many of its endophenotypes. And so we asked, first and foremost, we started with schizophrenia and asked, okay, schizophrenia, as far as we know, as far as we can tell, is not a disease of infraprimate mammals. We don't really have any evidence that there's a schizophrenic phenotype of any kind in monkeys. Some of the primatologists think that at the level of the great apes, there may be some psychotic disorders. And that pretty much maps to where human astrocytic evolution really takes off. So the question is schizophrenia glial disease. And this speaks to a larger issue, because we have lots of diseases in neuropsychiatry and neurodegenerative diseases both, where the operative word is neuro, and we just think of them as neuronal disorders, and that's largely because we've never had the tools to distinguish what was neuronal, what was glial, and that just led to assumptions being made. And so this speaks to the larger issue of what are the role of glia in these disorders, in neuropsychiatric and as the case may be, degenerative disorders. Is there more of a pathologic role for glia in the neurologic diseases? And so this is the experiment we did. We took patients with juvenile onset schizophrenia, and this was work done with Paul Teaser at Case Western, Rob Findling at Johns Hopkins. And Findling in particular has a prospective series that he's followed for a number of years of kids who were the children of parents with very strong family histories of schizophrenia. These are random genetics. The kids are all different genetically in this specific series I'll show you here. But what they all had in common was one side of the family with high penetrant schizophrenia. And the kids were therefore followed prospectively since they were at risk, and many unfortunately did become quite ill themselves. And these were childhood onset schizophrenics. Some of these kids as young as 8 years old, and all through the age of about 15. So we took 8 schizophrenics, and then roughly all gender matched and roughly age matched. You can't take control samples ethically on 8-year-olds, and so some of the controls were a little older, but as to the extent possible, age and gender matched, and then generated glial progenitors from them. Of course, we took skin samples, made IPS, made the glial progenitors from the IPS. And then looked first and foremost at the chimeric brains, just structurally, and saw that in control glial progenitor grafts in normal chimeras, and these were initially in shiverers, so hypermyelinated, and then the human cells take over and go ahead and myelinate. Now we're looking here at the cell distribution, not a myelin yet. And we see in the normals, the typical in these sagittal sections kind of architecture of the corpus callosum, the fimbria, the fornix over the hippocampus here. Normal pattern of glial progenitor expansion. The human cells are expanding throughout the white matter tracts and now starting to migrate at this point, about 5 months out, into the gray matter. Then we look over at the schizophrenia-derived. It's a whole other story. Much less expansion within the white matter. The white matter is not so prominent, but earlier and more significant expansion out to the gray matter, to cortex and subcortex both. And so the prediction then would be, well, gee whiz, there's something happening at the level of migration or differentiation so that the cells aren't expanding within the white matter the way they should. If there's fewer white matter progenitors, there's going to be less myelin. Well, in fact, in the prospective study with the kids, and what's really turned out to be a significant finding across the board in juvenile schizophrenias, is that these kids are all hypermyelinated. In fact, long before they're clinically symptomatic, by MRI, they have significant hypermyelination. Turns out that the animals are replicating that. So here's the normal pattern of myelin now in one of these chimeras. This was these cells were derived from a 20-year-old normal, and here were again the callosum, fimbria, fornix, back over the hippocampus here. We look at the schizophrenia derived, in this case from a 15-year-old. Well, first of all, it's very patchy white matter, but very little back here over the hippocampus, very patchy in the fimbria, and then you look at the high power. This is the normal pattern of myelin, and then here's the schizophrenia derived. It's very hypermyelinated. Many of the cells are not going ahead and becoming oligodendrocytes. Those that are doing so a little later with less myelin being produced. So we became really interested in this and thought, okay, here's a myelin defect, and actually we start to think then about schizophrenia as a myelin disorder, which is not something we'd really considered before. So we asked, well, what's the basis for this? And so we took cells and, of course, generated glial progenitors in vitro, and then but both from schizophrenics and controls, and then pooled in this particular case. This one we published last year. Took basically 4 different schizophrenic patients versus pooled controls. And this is all RNA-seq, looking at differential expression analysis, and you see many genes that are downregulated only a few that are upregulated, many that are downregulated in the across the board in these schizophrenic lines. By gene ontology, the major functional categories fall into synaptic transmission or glial differentiation. And with glial differentiation, of course, breaking out to myelination, oligodendrocyte differentiation, et cetera. So those are the two major categories. And then when we look at the actual genes involved, so first of all, the glial differentiation genes, without going into any detail here, but this is a who's who of genes that are involved in normal glial differentiation, normal oligodendroglial differentiation in particular. You've got LINGO, OLIG1, OLIG2, down here zinc finger 488, somewhere GPCR17 is down. So all the genes that we think of as canonically involved in glial differentiation are downregulated in the schizophrenics versus controls. And then, pari passu with that failure of differentiation, there are many both potassium channels and some of the glutamate receptor subunits that are differentially downregulated, but the real focus here is on potassium channels. Many of them are downregulated. And that corresponds to some of the functional defects of these animals. And then when we looked at those transcripts involved, at least annotated as involved in synaptic transmission, actually quite a few were down in the schizophrenic, including genes like nerexin, which is one of the canonical genes that's associated with schizophrenia. It's one of the few genes that is monogenically associated with schizophrenia. And in fact, previously it had been thought of as just a neuronal gene. Now what we realize is it's largely a glial gene, and it's the glial cell that's providing nerexin to the synapse. In any event, these are all downregulated, and we think they're downregulated secondary to the failure of glial differentiation. One way to test that is to see, well, if we take these cells, schizophrenic versus control, and we put them back into baby mice and chimerize them, do the schizophrenia-derived glial progenitors mature less effectively in the chimeric brain? And so here we've done that experiment, control-derived versus schizophrenia-derived glial progenitors injected neonatally. And these were injected into baby mice with the purpose of looking at the rate of astrocytic differentiation. We already know that doing the same thing, these would be hypomyelinated. Now the question is what's happening to the astrocyte, because the astrocyte and the oligodendrocyte, remember, are both coming from that glial progenitor. And most of the glial differentiation genes that I showed you that were downregulated are upstream of that fate choice. Okay? So genes like OLIG1 and OLIG2 are upstream of the point where the progenitor makes that fate choice between astrocyte or oligo. So if they're down, by the same token that we have hypomyelination, oligodendrocyte differentiation failure, we should have astrocyte differentiation failure, and that turned out to be the case. And so here we're looking at the production of, in red, glial fibrillary acidic protein astrocytes from the glial progenitor in the normal, versus in the schizophrenic, the engraftment is fine. Every little yellow dot there is a human cell, but it's only a handful of cells that have started to really make astrocytes yet by this point. So that has real implications, because normally in development, the synapses are forming, axodendritic contacts are occurring within essentially a network of astrocytic fibers. Astrocytes are there first as synapses are forming. Of course, neurons are out first in ontogeny, but as fibers start to be elaborated by neurons, that's when glia are coming into the fray in terms of their own migration and differentiation. By the time synapses are forming, typically they're forming within astrocytic domains. The implication here would be that, well, in early development, when this is happening, the astrocytes really aren't on the scene yet. The progenitors are, but they haven't matured as astrocytes yet. The implication would be that, therefore, axodendritic contacts are forming more or less on their own and organizing on their own in early development. These cells do ultimately become astrocytes, but even when they do, even they're delayed, but they ultimately do catch up and become astrocytes, but then they were really abnormal astrocytes. And so here we've mapped a number at a more mature stage, controls versus schizophrenia-derived. These are in the chimeric brains. And this is the normal radial structure, okay, of astrocytes. They really do look like little stars with essentially a radial array of fibers. Then you look at the schizophrenics, and these have a much more fibrous appearance. And this is the important point, many regions within their domains don't have fibers. They have larger gaps in between fibers, and then some of their fibers are basically sticking together and not recognizing that they are essentially sister fibers. And so if one takes any one of these cells and then rotates it on one axis, there's very little open space that occurs as one rotates. If you do the same thing with any of these, you actually have large gaps that really appear with cellular rotation. That's called Fanon analysis when you quantify that. And quantitatively, these cells had markedly more what we call empty wedges, meaning open regions within their volume, which means that in the adult, there are regions of essentially neuropil that are not being covered by astrocytic fibers. The astrocytic fibers are not controlling those synapses. One would anticipate that may have functional implications, so we looked at the behavior of the animals. And they, in fact, have a significant behavioral phenotype. The schizophrenia chimeras are extremely anxious. This is an open, in the open arms here, in this elevated plusmaze test, is where animals will typically go. The normals will spend a lot of time in the open arms, just engaging their environment. They're curious. When animals are anxious, they go into the closed arms and hide, basically. And it's a standard test of anxiety in mice and rats. And you can see the schizophrenic chimeras spend most of their time really avoiding the open arms. They spend very little time in the open arms. In fact, some of them just refuse to go in at all. They're also relatively antisocial. They tend to avoid other mice instead of being curious by them in the 3 chambers challenge. And then if they're in an open field chamber, they tend to run away. They spend more time escaping stranger mice. And so that's just by way of example. And in this particular case, all these dots are individual mice, but all the cells are from one patient. And so it's one patient versus its control, and of course we've done that on a broad number now of patient-derived lines, and therefore lots and lots of mice that are individually generated from individual patients. But here, what we've done is pool the data across patients, and so it gives you more of an idea of how this plays out, accounting for patient variability. And what's remarkable is that the anxiety and antisocial aspects of things, the socialization testing and the anxiety testing, is very consistent. Every single schizophrenic patient-derived animal we've looked at has been deficient relative to its matched control. Okay, and talking about the patients now, the schizophrenic patients across the board, the mice generated from them show behavioral phenotype that's actually quite constant. And then there's more variability in some of the straight cognitive testing, like novel object recognition, Barnes maze testing. But the bottom line is that these animals, even for those tests, even where there is more variability from patient to patient, we still see, when we look at the overall groups, a significant behavioral phenotype. So, it's not to anthropomorphize it too much. You can't look at a mouse and say that it has specific features that are schizophrenic, but clearly they have abnormal behavior by virtue of these glial abnormalities. And so then we asked the broader question: is the hypomyelination of schizophrenia, is that more typical of neurodegenerative disorders? Schizophrenia is, if nothing else, a neurodegenerative disorder. Over time, patients become more and more hypomyelinated. They start to develop cortical atrophy as they become more and more ill. And so we've looked at a couple of other diseases. I'm only going to show you one, which is Huntington's disease. And so Huntington's, for those of you not familiar with it, it's the result of a polyglutamine expansion in the Huntington gene. And it's a monogenic abnormality, but for many years people thought it was going to be a simple disease to take care of because it was monogenic. Well, it turns out that one gene, when it goes bad, just screws up a wide variety of cellular processes, both in terms of the RNA being the transcripts essentially being toxic, but also the protein itself binding non-specifically, both cytoplasmically and in the nucleus. And it's basically wiping out whole networks, both at the transcriptome and proteome level. And so it's a very complex pathology. But the bottom line is that patients with Huntington disease become hypomyelinated. And in fact, the TRACK-HD study, which was a study that came out a couple of years back, where patients were at risk were followed prospectively for a number of years. As in schizophrenia, actually, the patients become hypomyelinated long before they're clinically symptomatic. So we went ahead and then set up chimeras with Huntington glial progenitors. And so these were glial progenitors derived from embryonic stem cells that were either had the Huntington mutation or not. These are all sibling cells. And so these are typically from IVF couples where folks at risk for Huntington's would undergo IVF. And of course, the ES and then blastocysts that were developed but by virtue of IVF. Those that had the Huntington mutation, of course, are ditched, but many of these couples were willing to donate normal embryos as well. And so that gave us a set of sibling pairs of normal ES and Huntington ES, and then the glial progenitors derived from them as siblings. It's the next best thing to isogenics when you get down to it. And then we established the glial chimeras with those. And we found that they, in fact, were hypomyelinated, that there was a significant myelin phenotype. And I'm skipping over all that to get to then what we did with that information. We then went ahead and did the same type of RNA analysis, looking at the differential gene expression by the Huntington versus normal glial progenitors, and found a set of genes that was downregulated. We looked at three Huntington patients versus controls. And when we look at the gene ontology, basically when you look at gene set enrichment analysis of these data, it's quite remarkable in that top of the list, synaptic transmission, neurotransmission, and then most of the other categories fall into the essentially the rubric of differentiation failure or failure of neuroglial maturation. But it's a similar set of gene ontologies that we saw with schizophrenia. So then when we looked at the genes, what became remarkable is actually it's a very, very similar set of genes that are shared by the glial progenitors that are deficient in Huntington's and those that were deficient in schizophrenia. So again, a who's who of genes involved in glial differentiation and myelination are downregulated in Huntington's versus control lines. And one thing worth mentioning that we'll get back to, as a consequence, we believe, of the differentiation failure in the glial progenitors in Huntington's, a large host of potassium channel genes are downregulated, and that's something that becomes very important functionally. One thing that happens in Huntington's patients tend to be, their neurons are hyperexcitable, and if anyone is familiar with seeing Huntington's patients, they're hyperexcitable by virtue of neurologic phenotype as well. Many will go ahead to have seizure disorders, but more typically, of course, the choreiform abnormalities represent extreme hyperexcitability within the motor networks. So in any event, potassium, one of its major functions, at least in regulating the resting membrane potential of in this case glia, is to permit efficient potassium uptake by glia from the synapse. And when potassium channels, and especially a number of these are inwardly rectifying channels that allow potassium to get in other words, allow it to be taken up by glia from the synaptic cleft, if those are defective, if they're downregulated, then interstitial potassium levels are high and neurons are then hyperexcitable. And so it's really remarkable that literally most of the potassium channels made by humans are downregulated in these glia. And it turns out it's a common upstream regulator, as you might expect. So in any event, we then went ahead and set up these chimeras, and we working on the same type of logic that we did with the schizophrenics, that if these cells are deficient at the RNA level in terms of glial differentiation associated genes, we're going to see a failure in glial differentiation, and when we do. So when we look at in this case, at myelin production, we see in the normal myelin production by 3 months. When we look at the Huntington-derived chimeras, we just don't see any myelin production. So that's why they're hypomyelinated, but at the same time, there's a real astrocytic defect as well. They're not undergoing astrocyte differentiation as rapidly as the normal. And it turns out that structurally it's a very similar story as with schizophrenia, where we have normal structures in the controls, but in the mutant Huntington expressing astrocytes, as they mature, these large gaps, large regions that are not being covered by glial fibers. So there's a common theme that's arising here. There's a final common pathway of glial differentiation defects that is affecting the essentially the astrocytic development of these brains and therefore the neural network formation within it. So we wanted to look a little bit at the mechanism of that, and then broke down the RNA data a little bit further. So when you see these glial fibers that are sticking together in some regions that are not covered, it brings to mind some of the transgenics that have been described in with knockouts of different protocadherin genes. The protocadherins are a large set of genes There are 4 major classes. They break up into over 40 individual genes, but what they do is essentially establish combinatorial codes by which neuronal fibers either stick together or not, self-recognize or not, adhere with homotypically with partner cells or not. They've never really been looked at in glia. Well, we looked at it here thinking that, gee whiz, this kind of images we were looking at in these tracings I just showed you are very similar to the kinds of tracings that have been shown in disrupted dendritic arborizations in protocadherin knockouts. And so we thought, well, maybe these are really important in glial cells as well. So it turned out that they are. There's a sharp down regulation of quite a number of the protocadherins in both Huntington's and schizophrenia-derived glial progenitors and astrocytes. And here I'm just showing you one example where we've taken Huntington's and schizophrenia, and we've looked in each case at those protocadherins that are down-regulated in the glial progenitor. And the remarkable thing is this is a large family of genes, and yet it's the same set of genes, protocadherin 11X, 15, 7, that's down 17 in schizophrenia and Huntington's. And so that struck us as really interesting, because here you've got this set of genes that's involved in essentially establishing self-recognition, or as the case may be, self-fiber repulsion, that is not only down in both the schizophrenia and Huntington's-derived glial progenitors and astrocytes, but it's the same partners. It's the same individual moieties that are down in these very, very, very different diseases, but they are down in the respect of being downstream to the glial differentiation-associated transcripts that are also common to the 2 diseases. So we took that one step further and then looked at promoter analysis. In this case, we're showing Huntington's data, but we used TransFact to then go back upstream to see, okay, what regulatory elements are common to these protocadherins, to those protocadherins that are down in both Huntington's and schizophrenia, to try to really come up with, essentially more larger commonalities, and found that there were a number of transcription factors that are hitting all these different protocadherins that are down, where the transcription factors themselves are selectively down in Huntington's and schizophrenia. And the ones that really look to be the most prominent and shared across both diseases, NKX2-2 and DMR-T2. And so NKX2-2 has really become a significant target for us, because now we see that its downregulation is responsible for an awful lot of what we're seeing in these. So I'm going to close with just a couple of slides here to kind of take the logic one step further. If we've got these cells that are deficient, they're deficient in a number of functional respects, their ability to form proper fiber arrays, to organize synapses, to take up potassium, and they're deficient because of upstream defects that are fairly broad. What's the easiest thing to do? Can we really hope to go after all these different processes with individual agents for therapeutic, for treatment, for therapeutic benefit, or might it be simpler to just replace the cells outright? And so we took that tack. And the same kind of logic we used in terms of remyelinating the shiverer brain a while back, well, now we're thinking about that from the standpoint of replacing astrocytes using the glial progenitor. And so we started that work with Huntington's, and specifically asked whether we could take a Huntington's mouse, the R62 mouse, which is a really significant disease phenotype. These animals usually die by 16 weeks, and they're quite sick. And we wanted to see whether we could achieve any improvement in their motor phenotype by injecting human glial progenitors into the striatum, recognizing that those cells, recognizing or hoping that those cells would replace the mouse population, and could we then get therapeutic benefit by simply replacing that glial population outright? And so we did it initially in the striatum alone, and we're really focused on looking at the motor endpoints. And here we're just looking at one of these chimeric striata. All the red dots are human. The mouse, of course, in blue, gives you an idea of just how extensively chimerized. And this is a dot map giving you an idea of the distribution of these cells. In this case, we've sorted the cells on CD44, which is a more astrocyte-biased. It's the hyaluronan receptor. It's more astrocyte-biased from this population, a little downstream of that CD140 phenotype. And so we convert one into the other in vitro using bone morphogenetic proteins. BMP4 in particular drives glial differentiation from the progenitor. That's what we then transplant into these R62s. And then we see about a month delay in disease progression on the motor side, which essentially confirmed our hypothesis. We're happy to see that. The surprise was that even though we only went into the striatum, even then we still got a couple-week increase in survival. And so it was really quite effective. And there was a partial and incomplete, but actually pretty significant restoration of striatal volume. Normally the striatum, the quadriputamen in the mouse, becomes quite small, involuted in Huntington's, just as it does in patients. And here was a roughly 50% rescue of the degree of striatal involution. And so that suggested that, gee whiz, these glial, that it works, that you can actually get glial replacement with a therapeutic benefit. And then we went ahead and did this study. This was actually a study done by a CRO because one of the funding agencies we were working with wanted to actually see this in a blinded fashion, because this was the strongest survival effect that had ever been noticed in this very severe Huntington line. And so this is a behavioral testing by Psychogenics. It's a company that does essentially blinded behavioral analysis as part of FDA submissions and the like. It's done as a CRO for independent analysis. And so these were animals that we sent them all coded, so it was done blindly. And the animals were tested either for cognitive testing or in neurologic motor function testing. They call them respectively SmartCube and NeuroCube. But in any event, every animal is a dot. And this is essentially set up as a principal component analysis. And what we're looking at is either the first or second standard deviation from the mean. And that's these concentric circles. That's what they represent. And what you see here is a significant separation of the glial progenitor transplanted animals from the untransplanted R62s, from the untreated animals. That's not completely, there's no by no means a complete restoration here, but basically there's a significant improvement in both cases. And this is just with striatal replacement alone, not with whole brain, whole neuraxis replacement, which is what we're doing now. The mechanism for this seems to be a restoration of potassium homeostasis by these cells. And so what we notice is that the animals stop seizing, and they became much less hyperactive, jittery, seizing, etc. And so at the neuronal level, actually we noticed that the inward rectification improved significantly, and that the individual neurons, all this being done in slice preps, that there was much less of a voltage change in response to single current injections. And so basically it's restoring potassium homeostasis. Here, Miken's group then measured using potassium microelectrodes the striatal potassium level in the transplant versus the untransplanted animals, a huge difference. And basically the transplanted animals, their potassium homeostasis is restored to normal. It's no different, the interstitial potassium levels from normal wild types. And so And this, we're talking maybe 0.6 millimolar or so of potassium, but that translates to 7 or 8 millivolts, and if you just plug it into the Nernst equation. And so basically these neurons are much less hyperexcitable now, and that's contributing significantly to the restoration of phenotype. We suspect that this is going to be very similar to what we may see in schizophrenia as well. And so this gets to the point of essentially swapping out the cells in order to restore significant aspects of physiologic function. And if it really is that straightforward in terms of replacing the cells as opposed to treating all of the individual abnormalities that accrue from the disease cells, then we can think in terms of replacing astrocytes and glial progenitors in much the same way as oligodendrocytes, and that led to the design of this clinical trial, which is the pre-IND is before the FDA now, but it's a very similar logic to what we're doing with progressive MS in terms of giving glial progenitors in an allogeneic fashion for the purpose of replacing the endogenous progenitor pool. In MS, we're doing that in order for those progenitors to go to hypermyelinated or demyelinated regions to become new myelin. In the neurodegenerative diseases, the point is for those cells to replace disease cells, and that's going to be a question of the relative competition between diseased and healthy cells, but to have those cells, then the disease cells, the host cells, replaced by the transplanted donor cells for the purpose of astrocytic replacement and recolonization. So here's the basic strategy again. Whatever the source, we take glial progenitors, sort them using different markers. And I didn't go into this in great detail, but we have different marker sets for pulling out cells at different stages of either migration competence or replication competence, expansion competence, or for that matter, even migration preference. But depending upon what cell type we then take, we can go into either, we hope, pediatric or adult targets. Over the years, every time I have to review this, basically we're adding disease targets because the scope of glial pathology is becoming more and more apparent. And so to these classic myelin disorders, now we can add not only Huntington's disease, where replacement, at least in a common sense level, may perhaps make sense, but schizophrenia, where it becomes much more provocative, and yet everything we've done in the lab now suggests it should be possible in terms of replacing cell populations in the psychiatric disorders, and also work I didn't talk about at all. We've been doing just over the last year in frontotemporal dementia. We see much the same in terms of the significant role for glial pathology. That has been reported in ALS, in amyotrophic lateral sclerosis. Frontotemporal dementia is essentially the cousin of ALS. There are many disorders where it can be the same gene, and in some cases FTD presents, in some cases ALS, in some cases both. But in any event, this also looks to be a disease complex that is largely glial in terms of its origin. And I'll close with that. This is obviously Copenhagen and not Rochester. And the lab is now really divided between the two sites, both in Rochester here and Copenhagen here. There are lots of folks in Micah Niedergaard's lab we've worked with over the years as well. Lots of collaborators across the board in the various studies we're doing, but then also a number of clinical consortia we've set up for the translational efforts that I've described. And I'll close with that. Thanks so much.
All your background is always in an immunodeficient animal, and I think you use the reg2 knockouts most of the time. And my question is two. From the animal experiments, have you ever tried some transient immunosuppression or other types of knockouts, and how that will affect into the patients? Do the patients need to be under some kind of immunosuppression?
Yeah. So when we first started doing this, we did an immunocompetence. And so just for those of you interested in the animal modeling side of things, you can do this in immunocompetence if they're injected very early, meaning just on the first day after birth. There's a discrete window of perinatal tolerization to self that occurs across mammals. In mice, it's very short. You only have about a day. And then after that, transplanted, especially xenogenetic cells, will start to be represented as, or recognized, I should say, as non-self. But at least if you get cells in that first day, and if the numbers of cells are not too great, because large numbers of cells, then it's kind of a bit of a bell curve in terms of differentiation rate, and you end up with some cells differentiating late enough to present late antigens after the period of tolerization has ended. But if you use low doses and do it on the day of birth, you can get away without having to go into an immunodeficient model. It was when we started to do the survival trials that we realized we didn't want rejection as an issue whatsoever. And we were doing lots of animals. We couldn't guarantee that they were all going to be injected on day 1 or day 2. And so we crossed Shiver and some of our other lines, in fact, many of our other lines, to immunodeficient backgrounds. To your question, will patients have to be immunosuppressed? The answer is yes. So for anything allogeneic, yes, we're going to have to immunosuppress patients. And that's, there are very standard transplant regimens out there now that have been out for 2 decades for systemic organs. And so, I'm asked that question often, but the bottom line is it's not as bad as it sounds to have to immunosuppress people, at least with the current generation of agents. So our protocol includes tacrolimus and mycophenolate, and a period of just steroid and anti-CD20 coverage at just around a couple weeks of graft. But then with 6 months of tacrolimus and then stopping that and going on to mycophenolate. Now that's a protocol that's been looked at in a number of settings now, which is quite effective systemically. There's very little experience in the brain with any of this. One thing that is out there, kind of competing issues. The old sense that there was some immune privilege in the brain that I think has largely been rejected over the years. The brain's not all that immune privileged, because the brain's always subject to incidents that can result in blood-brain barrier opening, edema, etc., where now you can have antigen exposure across to the systemic circulation. But that being said, there's data out of the fetal Parkinson's transplants, the fetal midbrain transplants for Parkinson's that were done years back, that argued with very, very long-term survival, 13, 14, 15 years out, with patients who were then autopsied when they died of other causes, and their grafts examined and found to have perfectly viable, still viable grafts. And those cases had been immunosuppressed for 6 months, and then immunosuppression had been stopped. And it looked like that's a workable protocol. There was a parallel trial that had gone on at that time where patients were not immunosuppressed at all. Every single one of them rejected. So there is a period of immune suppression that's required. What's not clear is that if one stops immunosuppression totally and then proceeds, if a person has, it could be something trivial. It can be a flu or an adenovirus. Every time we get a headache, if you have the flu, you actually have a mild encephalitis. Well, you've got blood-brain barrier opening up, and is that enough to allow presentation of antigen that will then result in rejection? Nobody really knows, and so we don't want to take that chance. So in our trial designs, we have patients going on to mycophenolate, which is a relatively benign immunosuppressant for long period of time.
I have a question about neurodegenerative diseases that involve gliosis. In particular, in spongiform encephalopathy, prion disease or Creutzfeldt-Jakob disease, gliosis is a hallmark, and one of the ways to figure out, does this person have CJD? And in a mouse model of prion disease, a collaborator of mine has a PRP mutation where the spongiform encephalopathy happens spontaneously. When we looked at RNA-seq data from hypothalamus from these mice, demyelination, myelination was the process that popped up once you set aside the inflammation. Yeah. And there's also peripheral demyelination in humans that have the disease. So I'm wondering, have you thought about modeling and or therapy in diseases such as this, where gliosis and the demyelination are hallmarks?
Yeah, so couple of points there. Gliosis is a hallmark of all sorts of diseases of the nervous system. Anything where you have a secondary, anything that with a significant inflammatory stimulus in the brain, you're going to get gliosis. So it's a question really of whether the gliosis is part of the underlying pathology as opposed to a secondary response. In the case of CJD, we're very much interested in that because one of the aspects, of course, of CJD is that depending upon the prion mutation, you either get transmission or not, and some of that is, a lot of it is species-specific, right? That's why the different prionopathies have such tight species specificities. So the jury's out on that one. Actually, we're working with Stan Prusner's lab to model exactly this. They've set up the chimeras and have done the prion exposures there in San Francisco, and actually they're set up. We're just waiting for the results at this point to see whether or not this provides a useful model of CJD or the prionopathies more generally, and in a disease that frankly I'm more interested in from that standpoint, the multisystem atrophies. And so multisystem atrophy is very much a disease of glia as well. This is we used to think of this as Parkinson's variant or in some cases variants of frontotemporal dementias, but it's turned out it's a discrete set of disorders that in which glia, and primarily oligodendrocytes, but also glial progenitors are hit. With essentially it's a proteinopathy, but it's hitting the glia specifically, and so there's a massive glial response that is on the order of what you'll see in CJD, but where the glial cell is essentially the transmitting cellular phenotype, and that is something also we're modeling now in the lab using the chimeras.
I have two quick questions. Did you observe any cell deaths comparing patients with controls after transplantation? And did you investigate any local inflammation from microglia locomotion or around the area of the transplantation? And if it's different from patients and controls?
So again, this is an immunodeficient background. So for most of what I've shown you, just in some cases we've seen minor microglial responses. The microglia are still there. These are RAG1, RAG2, but nothing that's ever affected survival or viability that we've been able to tell. So we've not seen any significant cell death. We do see significant differences in terms of rates of expansion, depending upon, of course, donor and host, both, but not death per se. Okay.
I have a question about the migration and integration of the glial progenitor cells, and I wonder if you have any evidence that they tend to target pathological zones, like say in MS you have foci of demyelination,
yeah,
or
sure.
in Huntington's, or do they tend to more randomly integrate and replace existing glial cell?
Yeah, that's a great question. And actually, we've looked at it extensively because, to some extent, on a common sense level, we really thought we were going to see some sort of homing to disease sites, and we started doing that initially in the focal demyelinations, but actually in later work looked at some of the degenerative disease transgenics as well. And the bottom line is no, we see no homing. It's entirely stochastic spread, which makes it a lot simpler to model, if nothing else. The cells are expanding. So there's constant division of the progenitor population. There's contact-dependent inhibition of persistent replication. Once the cells, they're sending fibers out, and one of the things that these fibers are really queering are the presence of next-door neighbor fibers. And so the cells actually start to achieve. You can just imagine a balloon blowing up. Internally, as the cells that are the most dense, as they start to essentially recognize one another, they stop dividing. The cells at the margins keep on going, and there's an advancing wave of cells that are migrating and basically migrating rapidly enough that they're not recognizing one another. As soon as they become dense enough in a given region, the migration starts to slow, fibers start to recognize one another, they stop dividing, and they stop migrating. And so it's... from that standpoint, it's stochastic, but it's uniform, it's expansile, and it's predictable. We've even put cells right next to lesions, and it doesn't matter. So there's no homing per se. Now, remember, these are populations of cells that we're not exposing to any cytokine environment, et cetera, prior to transplant. The cells can be modified in ways that will allow specific cytokine recognition and allow following developmental gradients, and folks have played with those types of methods. But for everything I'm talking about here, where we're just transplanting native glial progenitors or ES-derived that are not otherwise manipulated, no, there's no homing that we've seen. So.